Hello, and thank you for watching this SoftGenetics webinar. My name is Adam Dillman, and today I'm going to be talking about the replicate comparison tool in GeneMarkerHID, which can be used to determine concordance of sample replicates. This is an advanced topic in GeneMarkerHID. To learn more about the basic operation of the program, please see the GeneMarkerHID introductory webinar. Forensic labs use replicates for many reasons, with the ultimate goal of deducing a consensus genotype. For example, they may use replicates to process multiple swabs of the same evidence source, or as insurance against reaction failure, allele dropout, and other common artifacts, or to process trace amounts of DNA. The replicate comparison tool in GeneMarkerHID assists with this process by automating replicate comparison, thereby improving the speed and accuracy of analysis. The analyst may compare replicates based on allele calls or additional parameters including size, height, and area. The user may simultaneously analyze up to 96 samples or 48 replicate pairs. Replicate groups of 3 or more are also possible. I'll now briefly demonstrate the replicate comparison tool. So now I've just uploaded a simple project into GeneMarkerHID. As you can see, I've uploaded 5 samples, each of which has 2 replicates, and I've labeled the samples 1 through 5, and then I've given each replicate the suffix of A or B. As we recommend in the GeneMarkerHID introductory webinar, I've already gone through and confirmed that my size calling was successful, and I've also addressed all flagging that I found in the allele report table. So I'm now ready to proceed to the replicate comparison tool to check the concordance of these replicates. But first I need to group the replicates together, and during this process I'm basically telling the program which samples to compare. To do this, you just need to go to Project, Apply Sample Grouping, and in this case obviously I want to group sample 1A with sample 1B, sample 2A with sample 2B, and so on. I have multiple ways of doing this. Right now we're being shown grouped by order, and my group size is 2. So the program is just putting the first 2 samples in a group, the second 2 samples in the group, and so on. And when I click match, you can see the result that's being shown, which is correct. In group 1, I'm showing sample 1A and sample 1B. In group 2, I'm seeing sample 2A grouped with sample 2B, and so on. Alternatively, you can click match by section, and here you can give section separators. So in this case, I'm using periods, underscores, things like that, to break my sample name down into different columns, and then I can use those columns to group my samples together. So in this case, in column 1 I have just sample, in column 2 I have my sample number, in column 3 I have my replicate identifier, and then in column 4 I just have a generic file name prefix. So in this case, by entering column 2 in my group identifier field, I'm indicating that my grouping information is in column 2, and that's correct because I can see my sample numbers there. So if I click match, you can see that I get the same result. Sample 1A is grouped with sample 1B, and so on. So there are multiple ways to group your files here. Once you have successfully grouped your files, just click OK, and now you can proceed to the replicate comparison tool, which is just under Tools, Replicate Comparison. This will open the replicate comparison main window, and the focal point of this window is the report table to the right. And just to orient you, we're being shown the details here for each replicate pair. So you can see we're seeing rep 1, the allele calls for allele 1 and allele 2 of rep 1, and then rep 2, and the allele calls for allele 1 and allele 2 of rep 2, and so on. And if we had a third replicate in this group, we would see 2 columns for the third replicate, and so on. We're being shown these allele calls for each marker, and each replicate pair is being identified by the first replicate in that group. Now you can see here to the right that we have 2 columns labeled status and final genotype. The status column is essentially showing you the results of the comparison of whichever replicates are in a specified group. So at a specified marker, you'll see a C if the replicates had the same call at that location. For example, here I can see that there's a C, And I'll also mention now that you can just double click on any point in this table to be taken to the corresponding electropharogram. So for example, by double clicking this C, I'll be taken to this relevant portion of the electropharogram. So I can see that sample 4A had calls 6 and 7, sample 4B had calls 6 and 7, and thus the status of this comparison is C. And C represents concordant. D represents discordant, meaning that the calls were not the same. So for example, if I click on D for D3S1358 here, I can see that sample 4A had the calls 1617, whereas sample 4B had the calls 1517. So obviously this is a discrepancy and the program is flagging it with a D, which again stands for discordant. If there's only one call for a group of replicates, and by that I mean if only one replicate had any calls and the other replicate failed, in that case you would see an N, which stands for null. And in null cases and C cases, the final genotype column will automatically be filled with the consensus genotype. So for example, if we look at all of these C calls, rep 1 for D21S11 had called 30 and 33.2. Rep 2 also had 30 and 33.2, and so the final genotype in this case is 30, 33.2. The same is true for every other C call, and the final genotype column is automatically filled in. For D calls, the solution isn't necessarily so obvious, and for that reason, we've given the user the ability to select the best call in their estimation from a drop-down menu. So here, for example, I can see that sample 4A had calls 10 and 11, whereas sample 4B had calls 10, 11, 12, and 13. So it's possible in this case we may be dealing with some contamination. So in this case you could choose to make the call 10 or 11, you could choose to make the final genotype call 10, 11, 12, 13, or if you felt there wasn't a consensus genotype available, you could select none. And also, after selecting a final genotype for a discordant call, you'll notice that the status column changes to E for edit. So this is a way to know if the call you're seeing in the final genotype column has been determined by the program or determined by a user editing the call. And you can only edit calls for discordant alleles. If we just look at another one here, for example, I'll click this one. Again, we can see that sample 4A had calls 18 and 19, sample 4B had calls 16, 18, and 19. So in this case, again, you could choose whichever call you felt was most appropriate for this location. I'd also like to point out that this report table has a filtering feature where replicate groups with discordant calls are filtered to the top. So in this case, you can see that when we opened this window, we can see all these discordant calls here, but if we scroll down to the bottom of the table, we'll see all of these replicates, all these other replicate groups are completely concordant, and we again can just check that by clicking on the status column or by these allele columns and just confirm that yes, this has 10 and 11, and the corresponding replicate also has 10 and 11. And as I mentioned, that's because there's a shuffle feature here where you can sort the table by status or by group sequence. And in this case, sort by status is the default, and that just pushes replicate groups with discordant calls to the top of the table so that they're easier to find. And that's actually really helpful if you're analyzing groups of 30 or more replicates. The ones with discordant calls get pushed to the top. You can bring your attention to them as quickly as possible. So here we've been comparing allele calls, but by clicking on this settings icon, you can actually choose many other parameters to compare between replicates. So for example, we could select things like size, height, score, if a given replicate has comments and the other one doesn't. There are a bunch of different parameters here that you can select. And you'll notice that if you select size, area, height, or score, you can actually give a threshold. So for example, if I select height, I can give a maximum allowed height difference between two replicates. So if I set this to 40% and click OK, you can now see that I'm getting more flagging because replicates are being compared by allele calls, and if their replicate peaks differ significantly in terms of height. So if I just click on this, I can see now that these two peaks differ by more than 40%. And so I'm seeing that flagging there. When you're done with your analysis, you can save or export your results in 3 different formats. We have the whole report, which just exports the entire report table. We have the final report, which only shows the status and final genotypes. And we also have the final report with only valid alleles. In that case, it gives the final genotypes and the status, but excludes any markers for which you've selected none. One last note, if you find that you have a replicate group that is just particularly discordant, for example, if one obviously shows signs of contamination, you can just disable the entire replicate by right-clicking and selecting Disable. And that'll exclude the entire replicate, and it won't be exported with your results. And for more information on the replicate comparison tool, you can consult the GeneMarkerHID user manual or visit the replicate comparison webpage on the SoftGenetics.com website. Thank you for watching this GeneMarkerHID webinar. For more information, or for a free 35-day trial of GeneMarkerHID and other SoftGenetics products, please visit SoftGenetics.com or email info@softgenetics.com. For technical support questions, please write to tech_support@softgenetics.com. Thank you for watching.